Hello everyone, in this video we're going to get into organic compounds. So we just talked about inorganic compounds, let's get into organic compounds. Organic compounds typically consist of groups of carbon atoms that are going to be covalently bonded to hydrogen, usually oxygen, and sometimes other elements as well. The four types of the most important organic compounds to human structure and function are carbohydrates, lipids, proteins, and nucleic acids. And so we're going to get into all of those today. Before we get into those compounds, though, you're going to first need to understand the chemistry of carbon and why it's so special and important for life. So if you take a look at carbon, you can see that it has 4 valence electrons. Remember that the octet rule dictates that atoms tend to react in such a way as to complete their valence shell with 8 electrons. So if you've got 4, carbon needs 4 more in its valence in order to be stable. Remember also, carbon isn't going to donate or steal electrons and make ionic bonds, but rather it's going to share electrons, forming covalent bonds. Carbons tend to share electrons with other carbons, and that'll often form long chains like you see here, a long carbon-carbon chain. They never exclusively just bond to other carbons, so they often bond to hydrogens as well. When you see a long chain of carbons bound to hydrogen, we call this hydrocarbon, like a hydrocarbon chain. Carbon can be combined with a number of different atoms, perhaps oxygen, nitrogen, or other atoms in a particular region of an organic compound, and sometimes the atoms that carbon bonds with may also be part of a functional group. Let's look at some of those functional groups right now. So here are some important functional groups in human physiology. We have the hydroxyl group. Remember, we talked about hydroxyl groups when we talked about dehydration syntheses. We talked about how one hydroxyl group bonds with the hydrogen of another hydroxyl group, making a water molecule and sticking those two monomers together. Hydroxyl groups are polar. They're components of all 4 types of organic compounds that we're going to discuss. And like I just said, they're involved in dehydration synthesis and hydrolysis reactions. Then we've got a carboxyl group. We'll see carboxyl groups when we talk about amino acids. They're found within fatty acids as well. And one of the names of it is carboxylic acid, so it wouldn't surprise you to find them in many other acids. Then we have the amino group. The amino group means that it contains nitrogen. We'll unsurprisingly find them in amino acids, and those are the building blocks of proteins. The next one is the methyl group. That's a carbon bound to 3 other hydrogens. If we go back just to look here, here is a methyl group on the end of this hydrocarbon chain, and another methyl group on the end of this hydrocarbon chain. And the last one is a phosphate group. A phosphate group, unsurprisingly, has a phosphorus atom in it, along with 4 oxygens. Phosphate groups are found in phospholipids and nucleotides, and we're going to talk about all of these in the molecules that we see today. So let's first get into carbohydrates. Carbon's ability to covalently bond means that many small, stable organic molecules that might exist by themselves are also able to form larger, more complex molecules as well. These larger molecules are going to be referred to as macromolecules. So macro means large molecules. Whenever macromolecules are made up of several copies of like a single unit, we'll call the single unit a monomer, and if you put many of them together, we'll call them polymers. So, for instance, this is a monomer. It's just a 6-carbon sugar. If you put 2 of them together, we call it a dimer. Mer means unit, and di means 2. Mono means 1. 1 unit, 2 units, many units for polymer. Remember, this is done through dehydration synthesis, and we showed it just earlier once again, where you have an OH group of one monomer joins with the H of another. This reaction results in the release of a molecule of water. It'll join the 2 monomers together. You can split them up, the polymers, into monomers by doing hydrolysis. Remember, to lysis means to cut or to rupture, and you add the water molecule back in, it'll break the bonds via that donation of the molecule of water. So take a look at this diagram. This is an example of one of the first macromolecules we're going to talk about, the carbohydrate. A carbohydrate is a molecule composed of carbon, hydrogen, and oxygen, and they're often found in the same relative ratios, 1 carbon, 2 hydrogens, and 1 oxygen. So for instance, glucose we might know is C6H12O6. So for every one carbon, there's two hydrogens and one oxygen, C6H12O6. Carbohydrates are referred to as saccharides, a word meaning sugars. There's three forms that are important in the body: monosaccharides, the monomers of carbohydrates; disaccharides, or two monomers; and polysaccharides, are the polymers, and can consist of hundreds to thousands of the monomers. So here are some important monosaccharides you may have heard before. Glucose was one of the best sources of energy for our cells, fructose, galactose, and these two are pretty important. They're the 5-carbon monosaccharides, deoxyribose and ribose, and we'll get into those just a little bit later. Here's the important disaccharides. These are sucrose, commonly referred to as table sugar; lactose, which you commonly might hear called milk sugar; and maltose, or malt sugar. Notice that they're both disaccharides, but they're disaccharides combined of two different monomers. So glucose and fructose makes sucrose. Galactose and glucose makes lactose, and two glucose molecules make maltose. So if you consume sucrose, your body doesn't just get energy by breaking down sucrose. First, you have to do a hydrolysis reaction to cut up the sucrose into glucose and fructose, and then your body can break down the glucose itself. Here are some important polysaccharides. Like I said, polysaccharides contain a few or 1,000 or more monosaccharides, and there's three important ones in the body. Starches, which are polymers of glucose, which just means many, many, many glucose molecules, they occur in long chains called amylose, or branch chains called amylopectin, both of which are stored in plant-based foods and are relatively easy to digest. Or maybe you've heard, eat a lot of starchy foods; that helps digestion. Glycogen, that's the next one. It's a polymer of glucose, but it's stored in tissues of animals, especially in the muscles and liver. It's not considered a dietary carbohydrate because really very little glycogen remains in animal tissues after they're produced for food eating. However, the human body will store excess glucose as glycogen in your muscles and liver. And the last one is cellulose, which is a polysaccharide that is the primary component of the cell wall in green leafy plants. It's the component of plant food referred to as fiber in humans. So having a diet high in fiber is something that people will tell you to do. In humans, cellulose and fiber is not digestible. You can't break it down. But like I said, it has a lot of health benefits. You can feel full, so you eat less. It'll promote your healthy digestive tract. And it's thought to reduce the risk of heart disease and maybe some forms of cancer. So cellulose is really important. Now, other organisms can digest cellulose. We can't. So just an interesting fact there. Although most body cells can break down other organic compounds for fuel, like fat and protein, all body cells can use glucose. And more importantly, nerve cells, the neurons in your brain, spinal cord, and throughout the peripheral nervous system, as well as your red blood cells, can only use glucose for fuel. So in the breakdown of glucose for energy, the cell makes this energy dollar ATP. ATP will release free energy when its phosphate bonds are broken, and we'll see that at the very end of this lecture. And that energy that's released supplies energy to the cell for the cell to do its own work. Here on this slide is the chemical formula for the breakdown of carbohydrate to produce ATP. You make much more ATP in the presence of oxygen, that's called aerobic respiration, than you would if you don't have oxygen, which is anaerobic respiration. And you realize what anaerobic respiration is if you've ever felt your muscles start to burn when you've been working out for some time. But this is aerobic respiration, and it's how the cell makes ATP, which is the cell's energy dollar. We'll talk about ATP, like I said, at the end of this lecture. Okay, let's get into the next molecule, the triglycerides. This is a lipid, so we're going to talk about lipids for the next couple of slides. One type of lipid is the triglycerides. A triglyceride is one of the most common dietary lipid groups, and it's the type most abundantly found in body tissues. This is fat, basically. It's formed from the synthesis of two types of molecules, one glycerol and three fatty acids. So you can see it on the right here. One glycerol, here's a fatty acid, You bind 3 of the glycerols to, look, there's 3 hydroxyl groups, and you get a triglyceride. 1 glycerol, 3 fatty acids. Let's see. A fatty acid that has no double carbon bonds anywhere along their length and therefore contains the maximum number of hydrogen atoms, these are called saturated fatty acids. And think about it like this. It's not quite correct what I'm about to say, but it's kind of a trick that I like to use. If it has all of the hydrogens and hydrogens are in water, then it's saturated, right? Like it's almost like wet. And I don't know if that'll help you, but it helps me. So if there's no double bonds, like we see here, a double bond, and when you have a double bond, you don't have as many hydrogens. If you wanted to count them out, you would know there's less hydrogens here than there are here. Then you're saturated versus unsaturated. When you have a saturated fatty acid, they're straight, rigid chains, and they can pack tightly together. And since they can do that, they're going to be solid or semi-solid at room temperature. Some good examples are butter and lard, right? If you take butter out of the fridge, it doesn't just melt into a soup, right? It stays relatively semi-solid. So butter is a saturated fatty acid. But in contrast, fatty acids that do have that double carbon bond get this kink, right? This is like it's kinked, like you bent it. And when it's kinked like that, they can't pack tightly on each other, and they're going to be liquid at room temperature. And then you might get some that are polyunsaturated fatty acids, which just means that they have 2 or more double bonds here. And when you get that, when you get a monounsaturated or polyunsaturated fatty acid, they're going to be liquid at room temperature. Some good examples of that are plant oils, such as olive oil. They'll typically contain both mono and polyunsaturated fatty acids. When we look at triglycerides, they're going to be a major fuel source for the body. When you're resting or you might be asleep, majority of the energy used is this fat store, the triglycerides, you're going to be storing in your fat tissues. Triglycerides also fuel long, slow physical activity like gardening or hiking, and they'll contribute a modest percentage of energy for vigorous physical activity. So when you're sprinting or something, maybe you're going to first be using the glucose because it's the most readily available energy source. But if you're doing something for a long, proactive period of time, like hiking, you're going to start going into your fat stores. All right, let's keep moving. The next the next lipid is the phospholipid. A phospholipid is a bond between the glycerol component of a lipid and the phosphorus molecule. In fact, phospholipids are pretty similar in structure to triglycerides. Take a look. There's the glycerol and there's 2 fatty acids, except there's not 3. The third binding group gets this phosphate. Gets this phosphate. In fact, instead of having 3 fatty acids, it gets that, it's called a diglyceride, but it's just a glycerol with 2 fatty acids and then that phosphorus group. What you see here is that this phosphate group is actually slightly negatively charged. And when it's slightly negatively charged, it becomes what is called hydrophilic or polar. And when things are polar, they are hydrophilic, meaning they are water hydrophilic, philia, loving. So the water molecules would like to be around the polar head of this molecules. And over here we have the hydrophobic tails. These are just long carbon, hydrogen, non-polar tails that do not like to be around water. And so what you get, it allows them to do something really cool, which is form the cell membranes of your cells. They do this in a double layer called a phospholipid bilayer. And we'll get more into this when we talk about cells, I think. But they orient themselves so all the water interacts with their polar head region because remember there's a charge on this phosphate, and all of the tails orient inward where there's no water. And so we'll probably talk more about that when we get to cells, but I just couldn't help myself. I'll talk about it now. Let's see, we'll talk about steroids, and then I'll pause this video so it's not too much longer, and then we'll do another video to finish out the rest of the organic molecules. But a steroid, commonly referred to as a sterol, is made up of 4 hydrocarbon rings bonded to a variety of other atoms and molecules. It can be bound to anything, but what you always see is 4 hydrocarbon rings as the main structure. And here's a cool trick for you if you're ever given the structure and saying, what is this? Well, this kind of looks like an S. It's got 4 rings that kind of look like an S. It Steroid starts with an S. I'm just trying to give you some tricks, and this is the one that I use. Um, so the really common one, and the one that I put here, is cholesterol. It's synthesized in the liver in humans and animals, and it's also present in a lot of animal-based foods. Cholesterol is really important because it's a component of our bile acids, which helps emulsify fat. Cholesterol molecules are also found in the cell membrane because, like phospholipids, they also have hydrophobic regions and hydrophilic regions. And what happens is cholesterol that might be in the cell membrane helps regulate the flow of substances into and out of the cell. Okay, so with that, I'm going to stop this one here. We'll pick back up on our next slide in the next video.